Assalamualaikum. Welcome back to my video. Let's continue on lesson learning number 5, which is DNA replication. So in this topic, we will discuss on the detailed process of DNA replication. So in DNA replication is a process of copying a double-stranded DNA strand. In previous video, we already learned the key important player in DNA replication, which is the DNA, the molecular structure of DNA. Now, we will take a look at detailed steps of DNA replication, and helicase, DNA polymerase, primase, ligase are important enzymes during DNA replication. So, you have already identified the type of model during DNA replication, which is the semi-conservative. Now, let's discuss more on this process itself. First, you need to know some of important terms such as DNA replication fork, origin of replication, leading strand, lagging strand, and Okazaki fragment. Those important words need to be kept in mind until the end of this discussion. So, DNA replication work is a Y-shaped region where the parental strands of DNA being unwound. Origin of replication is a specific site that indicates initiation of DNA replication. Leading strand is the strand that can continuously synthesize. Lagging strand, discontinuously synthesized strand, or also known as Okazaki fragment. Okazaki fragment is a short pieces of strand that discontinuously synthesized. So now, in this diagram, this is the mechanism of DNA replication. You have two strands. DNA consists of two strands. They complement each other and antiparallel, which means they are not oriented in the same direction. So one strand is in 5 to 3 prime orientation, and while the other strand is in the 3 prime to 5 prime orientation. Because of DNA polymerase, which is the enzyme in DNA replication, only adds new nucleotide to the free 3 prime end of primer or growing end of strand. So the formation of both new strands is different. Okay, there is continuously synthesis mechanism producing leading strand, while discontinuously synthesis mechanism producing lagging strand. So continuous synthesis, DNA replication is progressing in same direction as replication fork, so forming leading strand, while discontinuously synthesis, DNA replication on this strand can only occur in segments away from replication fork and forming Okazaki fragment, aka the lagging strand. As I said earlier, these are some important enzymes that involved in DNA replication process. You need to remember all those enzymes. For example, helicase untwist the DNA double helix at the replication fork, topoisomerase relief overwinding of DNA ahead of replication fork, and so on. So you need to remember. Okay, this one is the overview process during DNA replication. So during DNA replication, both leading and lagging strand production occurs simultaneously. Once DNA helicase unzipping the bases between the two parental strands, primase creates RNA primer immediately on both strands here. Okay, then DNA polymerase 3 then adds new nucleotides to the parental strands. One important point that you need to know is that the direction of DNA replication only occurs at 5 prime to 3 prime ends. Thus, one strand moves toward replication fork, while the other strand will move away from replication fork. At the end of process, DNA polymerase 1 remove all the primer, RNA primer, because it is RNA, and replace them with a new DNA nucleotide. So the lagging strand that is in short pieces fragments called Okazaki fragment will be joined by ligase to produce continuous strand. Okay. So DNA replication consists of 3 important steps, which is initiation, elongation, and termination. So we will take a look on step 1 first, which is initiation. Okay. So DNA replication begins at a site called origin of replication, where the two strands of parental strand are separated, and at origin of replication, it will be opened up and replication bubble form like this. So in eukaryotic chromosome may have hundreds or even thousands of origins of replication, and replication proceeds in both directions from each origin until the entire DNA molecule is complete. Okay, next, at the end of each replication bubble, it is the replication fork, which is a Y-shaped region where new DNA strands will be elongating. So helicase is the enzyme that untwists the double helix at the replication fork by breaking the hydrogen bonds between complementary bases, and then single-strand binding proteins will bind and stabilize the single-stranded DNA that is used to hold the separated DNA strands in place, and then topoisomerase enzyme will correct the overwinding ahead of replication forks by breaking, swiveling and rejoining the DNA strand. Okay, that one is the initiation step. Next step is the elongation. How DNA polymerase 3 add new nucleotides at the parental strand. Okay, now primase enzyme will begin to synthesize the RNA primer to the template parental strand, and primer binds to the starting site of replication. DNA polymerase 3 then adds nucleotide to the free 3' end of RNA primer and continues adding the nucleotides one by one to the growing new DNA strand according to the base pair rules. And then DNA polymerase 3 elongates new strand only in 5' to 3' direction. So you need to make sure where is the 5', where is the 3'. So the leading strand can elongate continuously 5' to 3' towards replication fork. So this one is the formation of leading strand. The other strand, called the lagging strand, that synthesizes discontinuously, and the new strand move away from replication fork. So another segment of the lagging strand can be made in similar way. Another RNA primer need to be added in each segment of lagging strand. So these segments of lagging strands are called Okazaki fragment. And then all the primers will be removed by DNA polymerase 1. DNA polymerase 1 replace the primer with the complementary DNA nucleotide, and then DNA ligase enzyme will join all Okazaki fragments to form a continuous strand. So here is the formation of lagging strand, whereby primase synthesizes primer, and then DNA polymerase 3 add DNA nucleotide to the primer, forming Okazaki fragment 1. Until it finishes, another primer will be added, and then another DNA polymerase 3 will add a new nucleotide at the new segment, and then forming Okazaki fragment. And all Okazaki fragment will be joined together to form continuous strand by DNA ligase. Okay, the last step is determination for proofread sequence to ensure that all nucleotides are paired up correctly. A with T, G with C. No RNA primer. Okay, and then DNA polymerase will double check the sequence of the new strand to make sure the nucleotides are paired correctly. DNA can be damaged by exposure to harmful chemical or physical agents such as cigarette smoke and X-ray. It can also undergo spontaneous changes. Any mismatch detected will be repaired. So the nuclease enzyme will cut the damaged DNA strand at 2 points, and damaged section will be removed. And then DNA polymerase 1 will repair by filling in the missing nucleotides. And then last one, DNA ligase seals the free end of the new DNA to all DNA, making the strand complete. That's all for DNA replication process.